medical news, a potentially fatal disease unknown just months ago is spreading so quickly that doctors now say it's a national epidemic.
90% of the cases have turned up in New York and 6 other cities. It is spreading at an alarming rate.
Researchers offer little hope for future victims because no one knows what causes AIDS or how to cure it.
722 cases of AIDS have been reported in New York City since the beginning of the outbreak.
Almost half of those diagnosed so far have died. AIDS was once found only among sexually active gay men, but not anymore.
Researchers complain that very little money has been spent nationwide to understand it or to combat it. One reason for that, they believe, is that the primary at-risk population is gay men who don't carry a lot of political clout.
This epidemic is not going to remain localized in any one group. Epidemics affect people. We need funding to stimulate research to find the cause of this new disease.
Would you support a massive government research program against AIDS?
I have been supporting it. We have $100 million in the budget this year.
The National Academy of Sciences has issued a very stern warning on the subject of AIDS. The current efforts are described as woefully inadequate.
This report calls for the government to spend $2 billion a year on AIDS research and education. The panel urged the use of condoms during intercourse.
We hope it will make the risky situation completely safe, but I don't think there's sufficient research to be certain.
He was tall, dark, and handsome, and he had that name.
Word of Rock Hudson's death spread quickly this morning, and friends and associates rushed to his Beverly Hills home.
For the first time today, the government approved prescription sales of a drug to treat AIDS.
AZT is not a cure. AZT only seems to extend life for an average 9 months.
42,000 dead from AIDS. Where was the FDA?
We want more experimental drugs tested in more people as quickly as possible. People are dying, we're caught in the middle, and we don't know what to do except protest and protest and protest.
Fight back! Fight AIDS! Act up! Fight back! Fight AIDS!
I would like you to join us in an act of activism.
Paul Watford,
Michael Talbot,
Rodney Arthur,
James Mincy.
A quilt made by Americans across the country was unfurled.
He was diagnosed with AIDS 5 years ago when he was 13, one of the first children known to have AIDS at that time.
There's a real danger right now of complacency in the fight against AIDS, because a lot of people feel, well, gee, I'm not going to get it.
Because of the HIV virus that I have attained, I will have to retire from the Lakers.
Officials at the World Health Organization say that 4 million women will die of AIDS by the year 2000.
I found out that I had been infected with the AIDS virus.
I asked the Republican Party to lift the shroud of silence which has been draped over the issue of HIV and AIDS.
AIDS patients are not to be pitied. None of us want pity. We do want compassion. We do want understanding.
The Scientific Advisory Panel has recommended that the Food and Drug Administration approve a new drug called the most important advance since AZT.
It's a step in the right direction.
AIDS patients are living both better and longer. New York, which has the highest number of AIDS patients in the country, has shown a dramatic decrease in the number of AIDS deaths, a 30% drop.
We have a fresh reminder this morning that the AIDS epidemic is anything but history. A report from the United Nations says that despite promising new treatments, more than 33.5 million people are now infected with HIV, and that number is expected to keep climbing.
Despite what you may have heard, I am cured of the AIDS virus.
We can now say with some confidence that we have the scientific basis to bring an end to this epidemic. We're not sure when that's going to happen, but we know it's possible.
Few could have imagined that we'd be talking about the real possibility of an AIDS-free generation. We've got to keep fighting. Fight for every person who needs our help today, but also fight for every person who didn't live to see this moment. Keep fighting for all of them, because we can end this pandemic. We can beat this disease. We can win this fight.
From ABC News. I want to welcome you to this morning's panel.
I think you're in for a really fascinating discussion. This is about ending the epidemic, science advances on AIDS. 2013 marks 30 years since researchers first identified the virus that causes AIDS. And the statistics are really daunting. Since the early 1980s, more than 60 million people worldwide have been infected with HIV. Some 25 million have died. More than 1 million Americans are currently living with HIV. And each year, 50,000 people in the US are infected. 20% of them do not even know that they have the virus. New York City has the highest number of new HIV cases in the nation. This morning, we're going to be exploring what science is doing, some of the breakthroughs that you just heard about, some of the challenges that still arise. And we're going to be putting a face on people who are living with AIDS, what they're experiencing. The group of people that we have assembled here today are some of the world's leading experts on HIV. Our first panelist was awarded the Nobel Prize in Physiology or Medicine for his discovery of reverse transcriptase. He's the former president of the Rockefeller University and the California Institute of Technology. Now, he's the founder of Calimmune Biotech Company. He's working to develop innovative gene therapies for HIV. Please welcome David Baltimore. Next, he was one of the original members of ACT UP, and he's featured in a recent documentary film about it entitled How to Survive a Plague. He founded the Treatment Action Group and AIDSmeds.com, and he's spoken out about HIV and AIDS on CNN, Nightline, 60 Minutes, and the pages of the New York Times, the Wall Street Journal, and Rolling Stone, among others. Please welcome Peter Staley. She's a pathologist and immunologist who consulted on some of the first cases of what came to be known as AIDS. As professor at NYU's School of Medicine and director of AIDS research at the New York Veterans Affairs Medical Center, she's been trying to solve its medical mysteries ever since. Please welcome Susan Zolla-Pazner. And he's a professor of medicine at the University of California, San Francisco, and a senior investigator at the Gladstone Institute of Virology and Immunology. He's run clinical trials on AIDS drugs all over the world and was named one of Time Magazine's 100 most influential people of 2012. Please welcome our final panelist, Bob Grant. So thank you all for being here and all of you for coming out. There have been promising breakthroughs in the past year and also some setbacks that we're going to examine. But I want to start now with the beginning and explore some of your personal experiences during those early years when you became involved in the fight against HIV and AIDS. So Susan, to start with you, you've been on the front lines of this since the beginning. I want you to take us back to 1981, 82, and share some of your experience about how you got involved and what that was like.
So there was a telephone call that I got in the summer of 1981 from a dermatologist at NYU, Dr. Alvin Friedman Keene, who said that he had seen 4 young men with an unusual presentation of skin cancer called Kaposi sarcoma. And since I was running my research lab studying cancer and also clinical immunology lab at NYU, he asked me if I could look at the immune function of these 4 patients. And they all looked the same and very unusual in terms of the number of what we call CD4 lymphocytes, a type of white blood cell in the blood. Some of these patients had none of these cells, which is extremely unusual. By the end of 1981, we had about 20 of these patients. And when you do anything in the lab, you have to run controls along with your experiments. And so we were using blood from people in the lab as our normal controls, and we said, well, this isn't right. What we need, because we had realized that all these patients at this point were young gay men, we needed young gay men healthy as our controls. And so I contacted a physician here in the city who had primarily gay practice. And we got specimens from 40 of his healthy patients. And we did the same studies on them as we did on our Kaposi's patients. And I will never forget, because for me it was the most dramatic day of the last 32 years in terms of this epidemic. We looked at the results and we realized that a third of these healthy men who had no symptoms whatsoever looked exactly like the 20 men with Kaposi sarcoma. And we knew at that point that whatever was going on, and we didn't know what was going on, but whatever was going on, a third of the gay population in the city were subjected to it too. It's really an incredibly chilling moment, and my professional life, you know, began in a sense, and it has never stopped since that moment.
David, how did you get involved in HIV/AIDS?
Well, actually, it goes back to 1970, because I was then trying to understand how a cancer-inducing virus grows, and discovered that there was a characteristic enzyme in the virus particles which copied RNA into DNA, which, first of all, was revolutionary in itself. But what it did was to provide a marker for this class of viruses. Now fast forward to 83, 84, and it was using that as a diagnostic that enabled Gallo and Montagnier and others to find the HIV virus and to provide the explanation for what Susan described, which was the ability of this virus to wipe out the CD4 lymphocytes and to put patients in a totally immunodeficient situation. So I sort of accidentally found myself involved. And then in '86, I was asked to head the panel for the National Academy of Sciences to look at our research response to this. I led a group that asked for $1 billion a year in research and $1 billion a year in clinical investigations to move this problem forward. And that was many-fold what was being spent at the time. And we actually felt a bit that we were being audacious in asking for it, but in fact, within a couple of years, we were spending $1 billion a year. And I think we can say that today we understand this virus better than any other virus on Earth, because we've put these really impressive resources behind understanding it. Hasn't actually stopped the epidemic.
And we will get to that. Bob, you were involved from the beginning.
Well, actually, compared to the others on this panel, I would say I came in late. In 1983, I was a second-year medical student in San Francisco and Berkeley. And I knew I was interested in social medicine, how medicine could interact with society, and I was looking for a job. And I wanted to study heart disease among city bus drivers, but there was no work there. Somebody said, you know, study this thing called gay bowel disease. You know, there's money coming in to understand the epidemic. And so, you know, and there was plenty to do. And it was exciting work. Imagine me, a 23-year-old, trying to discover a career, trying to make a career. And this was something where, you know, they needed all hands on deck. I mean, I'd taken computer programming classes, and so they said, we need you. You needed epidemiologists, you needed virologists, and there was nothing more exciting than this. I mean, retroviruses had just been discovered, and this was one. And I got paid half-time to talk about sex. I mean, it doesn't really get better than that as a 23-year-old. So it really set the course of my career. There have been times when I've been discouraged. At the next phase of my career, I was told not to focus on AIDS because by the time I was fully trained, the epidemic would be over, and that was 1987. And to this day, I don't know whether the person giving me this advice was optimistic about the end of AIDS or pessimistic about how long it was going to take for me to be fully trained. But that is a commitment. I want to retire after this epidemic is over.
Well, and your work has taken you around the world, and we'll get to some of that in a little bit. But Peter, I want to bring you in. Your involvement in this came from a totally different approach. You're the one non-scientist here on the panel. Tell us your story, how you came to be involved.
Well, the virus very directly brought me into it. I arrived in New York fresh out of college in 1983, and consider myself kind of a case study in how many gay men were infected in 82 and 83 and 84 because of the silence of the epidemic in those early years. Arriving in New York, working on Wall Street as a bond trader, I was very closeted. I certainly wasn't hearing my mayor or my president or even a politically active gay community talk much about HIV/AIDS. I certainly didn't hear that by 83, upwards of 50% of gay men were already infected with this virus. They hadn't discovered the virus yet, obviously, but that indifference probably led to my infection. And I found out right after Rock Hudson died in late 85, when the country was in a panic. The test had just come out earlier that year. And so I was one of the first to find out before I became sick. And in that sense, I was lucky, and one of the reasons I'm probably here today. But I joined ACT UP soon after it was born in early 1987, as I got more and more frustrated about this silence that was happening around this epidemic.
Well, as I mentioned earlier, you're featured prominently in the film How to Survive a Plague. I saw this film last week, and it's incredible. I encourage you all to watch that. But let's look at a little clip from that.
Three major things that control NIH research: stupidity, incompetence, and greed. Those are the three major driving forces behind what's going on here and why there are no treatments out there for people living with this disease. In that building down that way, Dr. Anthony Fauci is deciding the research priorities for the National Institutes of Allergy and Infectious Diseases. We're down here because we think we should be deciding the research priorities, because these are the people who know what's going on, because they're dealing with it every day.
Taken down by the police at the NIH.
Much younger me.
Yeah, I think several of our panelists, we've seen younger versions. I'm glad there's no footage for me. Now, why did you feel those kind of protests were necessary?
Well, we were dealing with first Reagan and then Bush, two administrations that were came to power largely based on what was called the moral majority back then, the predecessor of today's Tea Party, if you will. And these were very conservative administrations that had been ignoring HIV/AIDS for years, and as Dr. Baltimore's panel pointed out, was spending very little money at the NIH on AIDS research. We had an FDA that was still probably the most immovable bureaucracy in DC as far as how it slowly it would approve treatments, and the deaths were just racking up. And so our backs were against the wall. We could either roll over and all die quietly, or we could try to guilt trip the country into doing something about what we were going through and saving some lives. So it was about putting AIDS on the national agenda. It was about making the country see what was happening and care, and then forcing the government to actually start spending major dollars on AIDS research. And in short order, that money appeared.
And you have a couple graphs that you brought. One dealing with the issue of stigma and who was getting AIDS.
Gallup has been asking a gay-related question to the American public that's very antiquated now in today's debate. But it's the one that they've been asking the longest, and so they continue to ask it so they can get this wonderful 35-year sweep. And it's basically about sodomy laws. That's why it's very antiquated. But it is, do you think homosexual relations between consenting adults should be legal or illegal? When they first started asking that in the late 70s, the numbers were about even in the 40s. But then, as AIDS became a story with Rock Hudson, and a national panic broke out, there was a backlash that started happening against attitudes against homosexuals in this country. You see that red line, which is the people thinking that gay sex should be illegal. That started to really soar. And it hit a peak in early '88 at 57%. And then in 18 months, between that '88 number and the next time they asked it in '89, it had the biggest move in its history. It dropped 21 percentage points. And the only thing that happened historically in the AIDS crisis at that moment was ACT UP bursting onto the scene. And we had the FDA demo. That was followed by the quilt. AIDS became a huge national story from that moment on for the next few years, with a series of large demonstrations. And they saw gay men and lesbians fighting back. It just completely changed their attitudes for a while against how they viewed us. And we successfully made them, they may not have loved us, but they did not like the fact that the government was ignoring us while we were dying. That hurt their conscience.
Let's pull up that funding slide and take a look at the same timeline.
This is the NIH AIDS research budget and how it started with just virtually nothing during the Reagan administration. By the time we got to Bush, we were getting up to $200 or $300 million. And then during ACT UP's first 5 years, and after David's report, we kicked out and rose quickly to $1 billion. And from there, it just climbed to now it's over $3 billion a year. And those dollars in this age of austerity, those dollars matter. Those dollars are what eventually helped us get the breakthroughs that we have today. And those dollars are ultimately what we hope can find a cure and a vaccine.
David, I mean, what else do you think contributed to that rise in funding? Was it all the work of ACT UP and the advocacy community?
Well, no, the scientific community also acted up, and not so loudly or obviously, but used whatever connections we had within Washington to impress upon the government the absolute necessity to do something. And we were all terrified about how widely this virus would spread. We simply didn't know how widely it would spread. Now, in Africa, in places in Africa, it has spread to 75% of the population. In the United States, it hasn't spread that widely, but I find it still amazing that for all of the information that everybody has about this virus and about protecting themselves against infection, we have 50,000 new infections every single year. So it is a problem for the United States, but it's a much bigger problem on a world scale.
Susan, in those early years you were doing research, what impact did these protests have on you? How did that affect your life?
Well, we were doing the work. We had started our research in 1981 with the first patients that we saw. The problem that we were having was that we couldn't get funding. So we applied for our first grant to study Kaposi sarcoma in 1982, and the people who review these grants in Washington didn't understand the impact of what we were seeing. So it was not until 1986 or 1987 on that slide, where the graph is almost still horizontal, that we were able to get our first grant. So, you know, if we had had glasses that could have seen into the future, we could have had a much quicker start on understanding this disease. We would have had 5 or 6 more years of progress than we've had.
And when did the relationship between activists and scientists mature? I mean, was it strong from the beginning that there was an alliance, or there was a transition?
No, one of the things that was very painful for me to see in the Surviving a Plague film is that the activists didn't understand that there was a very caring, a very active scientific community that was trying to address the problem. And there was very little interchange between that community and our community. That began to change significantly, I would say, in the late 1980s, when the activists really came to have a voice at the NIH in terms of funding and priorities for research dollars. So we were probably close to 10 years into the epidemic by the time that the two groups came together.
So with that research funding, there was some success, some limited success in the late 80s with AZT. Then in 1996, a real breakthrough with the development of highly active antiretroviral treatment, or HART. Peter, how did those drugs change your life?
Dramatic. I remember coming back from the Vancouver AIDS conference, where they, kind of put a stamp of approval that this was real, and all of us we had been through so many ups and downs and disappointments that there was still some suspicion, like, really? This is gonna change the map this much? But we all went on the treatments that had been on those slides. We all started Crixivan and 2 nucleoside analogs and switched to them. I know I switched the month I got back from Vancouver, and most of my friends did. And within months, all of us went undetectable, which was just extraordinary. And we saw the death rate in the US and most of the developed world within 2 or 3 years drop by 80%. So it was a game changer.
Bob, how do these drugs work, and are there any limitations on that?
Well, the drugs work in combination to block different stages of the viral life cycle. They block, many of them block the enzyme that David Baltimore discovered, reverse transcriptase. They only work in combination, and many of the drugs that, like AZT, that had been discovered in 1987, which had only prolonged life for 9 months, when used in combination with another class of drug called protease inhibitors, blocking another part of the viral life cycle, and another third drug could prolong life indefinitely. So it was amazing to see how incremental discoveries could eventually lead to the home run, which was prolonged life and restoration of health.
And you've done work to show that these can prevent transmission, not just treat people who have AIDS. Are they good enough to create an AIDS-free generation?
Well, that depends not just on the drugs. It depends on how willing we are to use them. And I mean that by very broadly. But yes, these drugs, when used and sufficiently to suppress the quantity of virus in the body, virtually eliminate transmission from people who are treated to their uninfected sexual partners. And more recently, we've even discovered that these drugs can prevent people from acquiring the virus in the first place, a concept that we call pre-exposure prophylaxis. So there's a pill for preventing acquisition of HIV, just recently discovered and approved by the FDA, highly effective, and we're just now starting to learn how best to use it and who wants to use it.
We're going to get into that. With these developments, for many people, HIV has become a chronic disease in the way that for some people diabetes is a chronic disease. But there have been some numbers that are a little concerning. The HIV infection rate went up 22% among gay men under 25 between 2008 and 2010. How big an issue, Peter, is complacency? A sense that this is no longer such a fatal disease?
It is the issue. It depresses me to no end that, while the drugs that came out in '96 were such a turning point and a huge victory and saved so many lives, it was a double-edged sword in the sense that it caused a lot of us to fall down in fatigue after, this 15-year fight. And want to put this as far behind us as possible. And that's sadly especially true for the gay community in this country and in many countries around the world, where they want to move on. They only want to talk about gay marriage, gay marriage, gay marriage. And you'd be shocked to realize how the gay leadership in this country, with our huge lobbying organizations and our large foundations, how token their efforts are at this point on HIV/AIDS. And it's resulting once again in a rising infection rate. So silence may not be equaling death like it did in those early years, but it is equaling a whole lot of healthcare issues, the biggest health crisis facing gay men today. It remains so. And we will see the next generation at their current infection rates of over 2.5% a year. These are the 20-somethings right now. If those infection rates maintain, 50% of them will become infected by the time they're 50. And why should we continue to have generation after generation of gay men in this country, have a life of dealing with healthcare issues and health insurance issues and popping $25,000 worth of pills every year?
We're going to explore those issues in our third part in much more depth, but I want to turn now to talk about some of these breakthroughs that people here are working on. And see whether the strategies are in place, whether the tools are there to truly end AIDS. But we also want to hear from voices of people whose lives have been touched by the epidemic. And so we asked AIDS activists and some long-term survivors to reflect on how things have changed and what the science has meant to them. Some of the people in this video are HIV positive, some are not. But they're all activists, and they've all been working to try and put an end to AIDS.
In the early days of AIDS, if you were diagnosed, it was a death sentence.
I'd see these amazingly beautiful men reduced to just walking skeletons.
1993 was the worst year. That was the year I lost most of my friends. One hospital visit to one memorial service, to a funeral, to a cremation service, to a hospital visit. One after the other after the other.
It's hard, people don't come forward, and I understand why. I didn't come forward for 10 years. I lived in absolute fear and horror that I would be found out.
Those of us who remember the 1980s still have the emotional scars of the death and the dying. And those scars may have hardened and healed in some respects, but they're still there.
And one day, my roommate showed me this lump that he had on his shoulder, and this is about 1981, 82, and I went with him to the doctor, and the doctor said, "You have AIDS." We didn't really know what to do or to say, and I said to him, "Well, can you give us a referral?" And he said, "There are no referrals." are you religious?
Sometimes you were taken to hospital, they'd discover that you had AIDS, they didn't want to touch you. I mean, there were times when people would die and be put in like black garbage bags. I mean, these are the sons of people. They were treated like garbage.
People have a harder time believing that you can get HIV from one act of unprotected sex. In my case, I think it was one, maybe two, so with a friend, a good person, someone I knew, someone I trusted, someone who didn't know that he had it.
It took me 3 years to confirm that I was in fact HIV positive because I didn't want to know. None of us wanted to know, because if you were HIV positive, it probably meant you were going to have AIDS and then die.
My boyfriend, Michael Galt, died of AIDS. Michael was on the protease lottery. His number wasn't called because it wasn't approved in '93. His number didn't get picked. He died. To me, that shows what the meds did. How if you weren't lucky enough to have your number called, you died. And if you were part of a trial for the protease or lived long enough to actually be somebody who could just get it at a pharmacy, you started to live.
I remember that period where friends who looked like they were going to die, suddenly the retroviral drugs started being available, and they went from being emaciated, sick, Kaposi's, everything, to actually clawing their way back into some kind of functional mode. So hallelujah, that was amazing.
And frankly, had I not been in that early clinical trial, I might not even be here today.
There's so many people who didn't make it, and I was right on that cusp of, had I been diagnosed a little bit earlier, I might not have made it.
People died back then so that others can live today, and those who live today need to know the sacrifice that those people who died made in the 80s and the early 90s so that their sacrifice wasn't in vain.
Well, we want to now turn to some of the exciting science that's going on, looking for the next big breakthrough. And we're going to look at three approaches: vaccines, gene therapy, and drugs. But I want to start with vaccines. At a memorable press conference in 1984, the Secretary of Health and Human Services, Margaret Heckler, predicted that an effective AIDS vaccine would be available within 2 years. We're still waiting. David, I want to ask you, why has it been so hard to create an effective vaccine?
Well, first of all, we always make fun of that statement of Margaret Heckler's, but she had history on her side. When we discovered that a disease was a viral disease, we usually knew how to make a vaccine. And vaccines came along pretty quickly once we figured out how to make them. And so we have vaccines today for mumps and measles and smallpox and chickenpox and polio and whatever. So some of us suspected that this wasn't going to be true for HIV. And the reason was because HIV is a chronic disease that the immune system can't control. If the immune system could control it, then we wouldn't have the problem we have. And that one fact led us to be suspicious that when you tried to make a vaccine, you were going to discover that the virus had already figured out how to be resistant to the vaccine, and that turned out to be true. And so this virus, and it's not uniquely true of this virus, but there are very few like it, has figured out a whole variety of fascinating ways. I mean, from a scientist's point of view, it's amazing to see what's evolved. To counter the immune system, to not be sensitive to antibodies, to not be sensitive enough to T-cell immunity so that T-cell immunity can be used to protect against it. And so we have been unable to make a vaccine. And we have now seen fairly large-scale clinical testing of a number of vaccine candidates at phase 3, which is when you really look for efficacy, and they have all largely failed. One was partly successful, and we're still trying to understand exactly why that was true, and to see whether we can learn something from that that'll lead us forward.
Well, Susan, your work has been around vaccines. What's your strategy? How are you approaching this difficult virus?
Well, I think it's very important for people to understand that there are many different strategies that can be used for making a vaccine. So it's not a matter of a silver bullet. We knew how to make a vaccine to polio, therefore we can use the same methodology. We can't. There have been 5 large-scale clinical trials of vaccines. 4 of them have not shown efficacy. 1 of them showed marginal but statistically significant efficacy. I like what you said before about the fact that incremental discoveries lead to the final home run. And incremental discoveries sometimes are labeled as failures. But an experiment isn't a failure if you learn something from it. And so, for instance, we had a failure with a vaccine trial, a third large vaccine trial, which was called the STEP trial. The STEP trial was a real blow to the field because in that trial, individuals who got the vaccine were more likely to get infected than the controls. I mean, that is the worst possible outcome that you can look for. But we learned something, some very important things from that trial. So it was a failure in terms of it didn't protect, it actually made things worse. But we've learned some very important things from that vaccine trial, things not to do again. You've got to learn from history. The 4th vaccine trial was the Thai trial. It was conducted in Thailand, and the results were unblinded in 2000 and published in 2009. And there was 31% efficacy rate. Now, 31% is not as good as we want, but it was the first signal that we had that we could prevent AIDS. And that's a trial that I was not involved in the clinical part of it, but I've been very much involved in the follow-up to it. And there had been a lot of controversy in the field. There still is a lot of controversy in the field as to which strategy we should use to make a vaccine. You know, should we use a killed virus vaccine? Should we use a subunit vaccine? Should we stimulate the T cells? Should we stimulate antibodies, et cetera, et cetera? And the Thai trial indicated that antibodies are extremely important in prevention. And that is something that is absolutely essential for a vaccine.
What are, there's a term, broadly neutralizing antibodies. Right. This is something that you learned from the Thai trial?
Actually, no.
Right.
To be a little bit controversial, I would say that the field of AIDS has become enamored with these broadly neutralizing antibodies. These are antibodies that are isolated from a very small number of HIV-infected individuals, and they are fantastic antibodies. They can stop the infection of most any virus in very small quantities. And the hope would be that we could develop a vaccine that would induce these broadly neutralizing antibodies. That's the hope, but I refer to these antibodies as Michael Jordan antibodies. They're just fantastic antibodies. But I don't think that there's going to be a vaccine that's going to turn each of us into Michael Jordan. In the Thai trial, none of these fabulous broadly neutralizing antibodies were induced, but we did get some significant protection, which I interpret that certain types of garden variety antibodies that theoretically we can all make may in fact be sufficient to protect us. And so a lot of the work that I'm doing is to try and design a vaccine that will induce antibodies that we can all make that will protect us, instead of trying to turn everybody in the room into a Michael Jordan.
Now, as you mentioned, there have been a number of vaccine trials that have failed, trials where people who received the vaccine actually had higher rates of disease. Bob, I want to pull you in and ask you, you've done work around the world. When you have a trial that fails in that regard, how do you go forward with getting people to agree and countries to agree to do the next trial? Does it become that much harder?
Well, I think you have to start very early on in helping people understand the scientific process. You go into these studies really not knowing what's going to work and what's not going to work, and you do come up to the plate hoping to hit a home run. I mean, I think every trial that we've done hoping that this vaccine or this pill or whatever we were doing was going to completely block transmission. But if you can walk away from that having learned something, as Susan pointed out, then that's a win. So I think that we try to do as much as we can, and we certainly need to do more to tell people what the scientific process really is. One of the things that I think that the history that's being told today about drug development and the role of communities that needs to be told worldwide is that drugs and vaccines succeed when they're developed collaboratively and when there's input from broad sectors of the population. The story that you're hearing is not widely known in other parts of the world. I think that most of the people we work with in Asia, Latin America, and Africa still have the impression that drugs come in a postal box, already developed, already effective, already ready to go. And that is the way that antiretroviral drugs came to them. They did not have the experience of helping to develop those, of the early disappointments and the eventual success. I think we need to involve people, communities around the world, earlier in the process of vaccine development and development of other preventives, so that they can see that sometimes, a lot of the time, it fails, but sometimes it succeeds, and it's only through failure that you eventually get to success. So I think that, essentially mobilization of these communities, authentic inclusion of people, participants as well as the people who represent them in the scientific process from day one, helps people understand when things don't go well. It's because you didn't know whether it was going to work. You didn't know how to make a vaccine, but you have to try anyway. And so when it doesn't work, it's not so disappointing, because they knew from the beginning that it was.
David, what do you see as the biggest unsolved part of the puzzle when it comes to making an effective vaccine?
We've got to find a way to immunize people so that they don't have to take an action in order to protect themselves. That's when public health works. That's how the mumps vaccine, the measles vaccine, all of those work. We don't even think about the fact that we're protecting ourselves because we've taken our shot and that was in our history. And we've got to find a way to make the equivalent thing for HIV. And as Susan said, we have these broadly neutralizing antibodies. And using those as a model for immunization is a very difficult concept. And anybody who's trying to do that today, and there are a number of people trying to do it, knows that it's about as hard scientifically as anything they've ever undertaken. On the other hand, those antibodies are sitting there. We can make them in whatever quantity we want. And more importantly, we can put the genes for those antibodies into viral vectors. These are viruses that have been gutted of everything.
We're going to get to that work next. Yeah.
All right. So should I shut up and wait?
Yeah, wait just a second. I've never told the Nobel Prize winner before to be quiet. It's really something here. Okay. Susan, what do you see as the missing piece of the puzzle to reaching an effective vaccine?
Well, I think the question is whether we can induce antibodies with a vaccine in a realistic way in a relatively short period of time.
In a reproducible way.
And in a reproducible way, yeah. Right. I mean, the RV144, the Thai trial was one trial. I mean, 16,000 people, incredible amounts of money, effort. Running these vaccine trials is horrifically expensive. and then you get a 31% efficacy rate, and you say, and there are a lot of people who say, well, you know, I don't quite believe this. I happen to believe it, but it's not easy to reproduce an experiment with 16,000 people in it, okay? And then times change and products change. But one of the points that I want to make, and this refers back to what David was saying, is, you know, you get 2 shots for mumps, I think, or for measles, right? You don't have to come back 2 months later, 4 months later, 9 months later, 12 months later, to get boosted and boosted and boosted. And the immunity lasts for a long, long period of time. The problem that we're having, even in the one successful trial that we have, is that it required 4 immunizations over 6 months. And 6 months after the last shot, all of your antibodies were gone. So you didn't have a lasting response. So what we need to do, the most critical thing, is figure out what the correlate is of protection, okay? I think that a major part of that is antibodies, but then you have to induce them with 1 or 2 shots, and it has to give a response that lasts for a long period of time.
Well, I want to shift now to what David was starting to talk about. Not everyone is working on vaccines. There's the area of gene therapy. Can you talk about what Calimmune, what your company is doing, how this approach is different and novel?
Actually, there are 2 things that we're doing. One involves Calimmune. The thing I started talking about doesn't.
Okay.
It's work that we're doing with the National Institutes of Health. And what we're trying to do is to design viral vectors that encode these super antibodies, these Michael Jordan antibodies.
What's a viral vector?
So a viral vector is a virus in which we have taken out the genes that make the virus dangerous, make it a source of pathology. And we replace those genetic regions with something that we want to use. In this case, the genes for these super antibodies. So we can design a virus that encodes really nothing but this antibody. And we can put that into the muscle of a mouse, and we turn the muscle into an organ to produce antibodies. And so we can actually get phenomenal levels, concentrations of this antibody made in a mouse after a single injection, and it lasts the lifetime of the animal, and it protects the animal against HIV infection for its lifetime. We have to do some tricks to get an HIV infection in mice, but I'll leave that aside. And so we're now taking this to humans. I mean, we're going into manufacturing so that we can try this in humans. There's really no reason why it shouldn't work. The viral vector that we're using has been tested in humans. It does lead to protein production. It's never led to antibody production because nobody's tested it. So we're actually pretty confident. Now, the Calimmune story is another story, and I'll leave that aside.
Okay. But this is going to be going into humans?
Within the next year or 18 months, I would say.
Peter, would you sign up for a clinical trial like that?
Yes. If it were, in my case, it would have to be if they tested as a therapeutic vaccine. But certainly, I would love to not have to take handfuls of pills twice a day for the rest of my life, and all the side effects that come along with that. Getting my immune system to fight this virus along with these antivirals, or in replacing these antivirals, is what we call a functional cure, and it's something I hope to be around to witness and to take.
You know, David, you've called the gene therapy approach a Hail Mary approach.
Yes.
Why go down this avenue if it's such a long shot?
The reason is because nothing else is working. If we had a standard way of making a vaccine, if Susan can figure out how to make an immunogen that will elicit antibodies that will protect people, I'd go out of business tomorrow, because what we're trying to do has not been ever done on a large scale in the human population. And so we don't know what we're going to run into. And I don't want to find out what we're going to run into unless it's absolutely necessary. But my judgment is right now it's absolutely necessary, because all the methods that we have for protecting against transmission require that individuals take action every day, every month, every week. And people just don't do that. And so they don't protect themselves in a solid way over their lifetimes, which is what they have to do. And that's what a vaccine does. That's why we're all here so comfortable these days.
The third approach to trying to knock out HIV is looking for a drug therapy cure. And when you think about bacterial infections, you get a pneumonia, you're given an antibiotic, and the infection is cured. And so the idea is, could drug therapy be used as a way of wiping out HIV? Currently, that's not the case. It keeps it at bay. But then it comes roaring back in most people if you stop the drugs. Why, Bob, is it so challenging to try and rid the body of HIV with drugs?
Well, there are several challenges. I think before we diss the drugs, though, let's emphasize that when started early, they restore the immune system to health, and people, as nearly as we can tell, can have either normal or near normal lifespans. These drugs, though, cannot be stopped, as you mentioned, because the virus is still there waiting in the wings. This is a virus that integrates itself into the human genetic material. So it has an integrase, an enzyme that gets the viral material into the human genome. And that allows for latency. It also allows for persistent infection in a variety of different cells, including a cell that we call macrophages, which exist in the brain. And it really exists in all of our tissues. But I think we're just now learning that the drugs also are not really penetrating as well into all the tissues of the body as we had hoped. We know that these medications can lower viral load in the blood to very, very low levels, but it doesn't eliminate them. But the drugs are not quite getting into all of the different parts that are infected, the part of the intestine, the part of the brain, and it does now look like there's ongoing viral replication occurring even when people are taking combinations of therapy. So that's the bad news. The good news is that some of these new drugs, some of the more recent therapies, do appear to be having additional benefits in decreasing or eliminating all signs of viral replication. Which can be quite subtle. We're not talking about viral load anymore, but we're actually talking about measuring the intermediate steps that the virus takes as it replicates. And we can see that use of some of the newer classes of drugs is causing those to decrease even further. And that's good news. It opens up the question again that therapy started early enough and using the right combination could be sufficient to reset viral replication. We also have some very promising cases. I mean, we've heard from a cohort in France of people starting therapy very early. Something like 15% of them can eventually go off therapy and their viral load continues to be low. So it doesn't happen in the majority, but it's 15% that appear to have some sort of durable ongoing benefit. And we think that that is partly due to starting therapy very, very early. And of course, the case that recently was described at the retrovirus meeting just this last year of an infant who started therapy within 36 hours of birth and continued on therapy for more than a year, then was lost to follow-up, came back in, and evidence that the virus is still in that infant is just very, very hard to find. And so it looks like that infant, because therapy was started so early and so intensively, may have had a markedly altered course of their infection. So we're more and more optimistic that therapy, especially using new classes of drugs, could really have a marked impact on the course of infection.
There are a couple of terms that are thrown around: cure versus functional cure. What does that mean?
Well, it's actually similar to what we've been talking about in cancer for a long time. I mean, there's in HIV medicine, just as in cancer, there's skepticism that we're ever going to get rid of every last cancer cell or every last HIV-infected cell. That may not be possible. But does it matter if someone has such little virus that they can go off therapy and the virus doesn't come back? It may still be present in 1 or 2 cells or a very, very small number of cells, but if it doesn't rebound, if it doesn't really cause injury to the immune system, then, and people are able to stop therapy, then that's a win. And that's what we call a functional cure.
And your work now is looking at the use of drugs, both pre-exposure and post-exposure. What are you finding? I mean, there's some exciting results from that. What have you found, and what are the next steps you're doing moving forward?
Well, the concept really is to use the same medicines that can prolong life in infected people to see if they can prevent the acquisition of infection in uninfected people. So that concept is sometimes called pre-exposure prophylaxis. The concept is that people can have a hard time protecting themselves on a day-to-day basis, but they often recognize that there's something about their lives which could put themselves at risk for acquiring HIV. And we now have a pill that they can take, recommended to be taken on a daily basis, that prevents the acquisition of HIV. We were able to demonstrate that this works for gay men and trans women. Other studies have shown that it works for heterosexual men and women as well. It does require that people take a pill every day. And we know that that is a challenge. But it certainly can be done. I mean, people routinely take a pill every day to prevent heart attacks or strokes in the form of medicines for blood pressure or for lowering cholesterol. We also know that pills can be taken every day to prevent pregnancy. So it is feasible for people to take a pill every day to protect themselves. And this is just another prevention pill, but this one is for HIV. Now, you know, so far, only a minority of people who are offered the pill say they want it or are willing to take it every day, but it's a significant minority, maybe 25, 30%. And we're finding as we're able to counsel people more clearly about the very excellent benefits that they get from this, that the percentage of people willing and wanting to do it is increasing.
What does the funding picture look like for funding around drug therapy, a drug cure for HIV? Is it where it needs to be?
Well, I think that just in the last year and a half, there's been new NIH initiatives to fund research on a cure, inspired in part by Martin Delaney, who was a key activist who helped the research community stay focused on a cure, realizing that therapy that prolongs life is good, but it's not good enough. Eventually, we do need a cure. And so I think Martin Delaney inspired a group of scientists to really propose a collaborative process for looking for a cure. Is it enough money? No, I don't think it is. I think that eventually a cure is the way that we can treat and prevent this disease. But I would also say that starting with the amount of money that they've given is a good first step, because we do need new concepts in cure research.
Yeah, it's interesting because we saw with the graph how federal dollars can dramatically change the course of an illness. It's the same thing with cure research. The dollars we spend now will ultimately lead to a cure. I'm convinced of it, but we have to make that upfront investment. And it's very so it's a debate we need to have, especially when we're getting pushed back, being told that we're in an age of austerity and can't afford things like this. It's very, very frustrating. But cure research is hot these days, and it's something we should be putting more dollars into. And it's a sea change from as recently as 10 years ago, where both AIDS activists like myself and researchers all didn't even consider it in the realm of possibility anymore. Kind of had given up hope on an ultimate cure. But with the Berlin patient, we have a person cured of HIV now. And we possibly have a baby that was cured of early infection. The the possibility, the resistance to the idea that it's possible has fallen by the wayside. Everybody thinks it can be done. It's just a matter of time and effort.
David, the Berlin patient, just want to touch on that briefly. A patient who had cancer had a bone marrow transplant and was then free of HIV infection. Is there anything to learn from that in terms of strategies moving forward? It's not an antibody approach, it's something different.
Right. So the antibody approach that I talked about is a method of protection against becoming infected. But the Berlin patient is a situation in which somebody who was infected was treated and the virus was eliminated apparently from his body. You got to keep saying apparently because it's very hard to prove a negative. All you can say is that there has been no evidence of virus in that individual. And the reason for it was that the individual was provided with cells that don't have the receptor for HIV. So for HIV to infect a cell, it's got to see the surface of that cell. And what it sees, among other things, is a molecule called CCR5. And so the Berlin patient was transfused with the blood of somebody who doesn't have CCR5 on his cells. And then those cells grew up in the patient, and now the patient has cells which are resistant to HIV infection. And the HIV infection went away. So you talked about Calamune. In this company, Calamune, what we're trying to do is to mimic the Berlin patient. But we're doing it not by finding people who are naturally resistant to infection, who are very rare, but rather by inducing resistance using what's called an siRNA. Again, it's a gene therapy style of approach in which we're putting into the patient's blood cell system a virus that encodes something that counters that receptor, doesn't allow that receptor to appear on the surface of the cell. And so it makes the cells resistant to HIV. And we're hoping that that will work, and that's going into clinical trials right now, and the first patients will be treated next month. And we're very hopeful that we can use that as a way of effectively curing patients and allowing them to live drug-free thereafter.
So seeing the benefits that the Berlin patient had, but without the risk of a bone marrow transplant and immunosuppression and the whole 9 yards.
Well, actually, we have to do some things that are not so terribly different, but we don't have to put in foreign bone marrow into the patient, which he had to do for the Berlin patient.
Well, all of the breakthroughs we've been discussing give hope that there could be an end to AIDS. And we're going to get to that topic now, but let's go back and listen once again to some of the voices of those who are in the middle of the fight.
My hope for those living with HIV and AIDS is that they all get antiretrovirals so they remain healthy, so they don't spread it, so they don't orphan their children. But my real hope is a cure.
I want to see that breakthrough happen. But I don't want the lessons that we learned in the old days to ever be forgotten. The biggest misconception back then was, and still is, it'll never happen to me.
So it's not the red flag that it was. It's something people have been hearing about for 30 years. So it doesn't have the resonance that it once did when it was the new disease of the century.
You have enormous scientific accomplishment. You have enormous scientific potential. That's the good news. The bad news is we have tens of millions of people not in care. We have lots and lots and lots of new infections, more than 50,000 a year in the United States of America alone, which is unthinkable and unnecessary.
I think people think it's gone away, but here we are with still 34 million people HIV positive in the world, and here we are, with this extraordinary opportunity where we can end it.
I know we're getting closer, and I hear about it, I read about it, it's talked about, it's exciting, we're getting closer to a cure. It's a worldwide pandemic, so there's a lot of work to do. And so the closer we get, there's years and years of work ahead in getting whatever breakthroughs we've got into the hands of everybody who needs it.
I have always gotten out of bed in the morning because I believed. I allowed myself to believe that we would cure it. If I didn't believe that, I would never get up. I said to my mom, what do we do if I'm cured? And she said, we move on to the next thing. We go help with another cause, help with another disease. But it would be an incredible thing to live to that day and to, sorry.
My hope that those of us who are living with the virus might someday have it eliminated from our bodies, and my hope that someday no one will be HIV positive.
A recent United Nations report said the end of AIDS is entirely feasible. And I want to get the take of each of you on that. Is that feasible? Is that something that we could see in our lifetimes? Susan?
I don't think we're going to see it in our lifetime or in my lifetime. I see a lot of young faces out there, and I'm hoping it will be possible in their lifetime. One of the things that worries me about the discussion about the end of AIDS is not that it's not down the road. I think it is. But that it is going to raise unrealistic expectations much too soon. We're going to have a vaccine in 2 years. Well, no. I think that people need to understand that there are going to be hills and valleys, there are going to be successes and failures, and it's the process, and it's going to take us a while to get there.
It's interesting to note that these phrases, end of AIDS and AIDS-free generation, did not really come from scientists. They didn't come really from the activists. It came from the politicians. Mm-hmm. And they're great. They are obtainable goals. We all believe that. But right now, they're political statements that aren't being backed up by the resources that we need to actually make it happen.
Bob?
Well, I'd like to be the voice of optimism. I do think that it's feasible. I think that we know how to end this epidemic. I think that getting it done, though, is not to be assumed. I think all of us will probably end up doing a lot of what we like doing and some things that we don't like doing. The scientific community needs to invest much more in what we call implementation science, how best to use therapies that we already have. It's not the kind of science that has excited, those of us on this panel, but it's valuable science, it's interesting, it requires a new skill set, a new generation of young people to take this on. And I think that kind of research, really how to implement things. I think making commitments to healthcare as a right it could be one of the most important legacies of ACT UP. Healthcare is a right. It's not okay to let friends and family or even strangers die of a disease that you could treat or that you could invent a treatment for. That principle has not been universally accepted around the world. In practice, we do not treat healthcare as a right, and yet I think deep in our hearts we believe it is. Because we can't stand by and watch people die of something that we know how to treat. And really establishing political will to make healthcare a right, just as education or other things, is one of the legacies of ACT UP that is required to really implement what we know how to do. It's going to take more money, but it's not an unmanageable amount of money. The amount of money we spend to keep ourselves safe from HIV is a small fraction of our total security budget. I think humanity has made it very clear that we will spend enormous amounts of money to stay and feel safe. We just need to define safety from HIV as part of that agenda, and we'll find plenty of money. The amount of money to fully fund AIDS programs around the world is small compared to what we're spending on other things.
David, is an end feasible?
I don't think an end is feasible if it requires action on the part of individuals to protect themselves. Because too many people grow up through adolescence when they believe that they're immortal. and they still smoke. I mean, for all of the years that we have been saying that smoking is a death sentence, people still smoke. Newly emerging gay men still have sex without protection. And so I think we have to see this as a public health problem. We have to find a way to protect people so that they don't have to think about their protection. And then maybe we can actually put the genie back in the box.
You know, even without a new tool for eliminating AIDS, there are a lot of tools right now that have changed the face of this pandemic. But I do want to pull up one slide here that looks at AIDS around the world and where AIDS is occurring, because it's not one pandemic anymore. It has a very different face in the United States to what it has around the world. And Bob, you were talking about implementation and money for implementation so that existing tools can get out there. The majority of disease in sub-Saharan Africa and parts of Asia. Is enough being done to implement what we already know and can use to prevent and control AIDS?
No. We continue to underfund the global effort on HIV. Estimates of what is required range between $24 and $35 billion a year, depending on how much of the existing portfolio you want to implement. My bet is that we're going to have to do all of it and do all of it all the time. So I would aim higher. The most we've been able to raise globally for the war on AIDS is about $18 billion, I think. So we're underfunding it, but again, not by an impossible amount. I mean, we could raise the additional money by making this a focus of human safety.
David?
I don't think we will eliminate it that way. We can help people. There are many people who would be helped enormously by availability of drugs today, but that's not going to put the genie back in the box. And I think we have to think of it that way. And I personally believe that the discussion about the cure for AIDS is a good thing, because it's focusing people on thinking about new ways of approaching the problem. And the one thing that we haven't had enough of is new thinking. And so I'm very supportive of that, but I don't believe it's going to happen on any kind of short-term scale.
Susan?
Well, as I said, I think it's a long road, and I'm very concerned about unrealistic expectations.
Peter, I want to close on something that relates to you. There's a very powerful moment in the movie How to Survive a Plague, and I want to show that and then ask you a question about that.
I'm going to die from this. This isn't going to be cured for years and years and years and years.
You were in your 20s when you said that. Do you still think that's true?
No, I'll probably get hit by a bus before HIV is going to get me now. And that's an amazing thing to be able to say. I'm 52. I'm dealing with aging issues now. Joy of all joys, right? And I, maybe I'm younger than you are, I don't know, but I think I am going to be around when they get the cure. I agree we have to lower expectations. It's not going to be within the next 5 years. Cure research is in its infancy now. But it's a hot area, and I think it's going to pay off in the end. It took us 15 years to get to the therapies that are saving my life now. It might take 15 years to get to the cure, but I should hopefully still be around then. So.
Can I just sort of bring things full circle by saying that when we saw our first patients in 1981, they were dead within 3 months. So we have lived through an incredible arc during these last 30 years, and now people are living with AIDS and facing the problems of aging. Never in my wildest dreams did I think 30 years ago that that would ever be a problem. So I think we can end on a very optimistic note about what the possibilities are and what they will continue to become.
I think that does bring us full circle. So I want to close and thank our panelists, David Baltimore, Peter Staley, Susan Zolla-Pazner, and Bob Grant for a really terrific hour of discussion.